Ku is a dimeric protein complex that binds to DNA double-strand break ends and is required for the non-homologous end joining (NHEJ) pathway of DNA repair. Ku is evolutionarily conserved from bacteria to humans. The ancestral bacterial Ku is a homodimer, two copies of the same protein bound to each other. Eukaryotic Ku is a heterodimer of two polypeptides, Ku70, XRCC6, and Ku80, XRCC5. So named because the molecular weight of the human Ku proteins is around 70 kDa and 80 kDa. The two Ku subunits form a basket-shaped structure that threads onto the DNA end. Once bound, Ku can slide down the DNA strand, allowing more Ku molecules to thread onto the end. In higher eukaryotes, Ku forms a complex with the DNA-dependent protein kinase catalytic subunit, DNA-PKcs, to form the full DNA-dependent protein kinase, DNA-PK. Ku is thought to function as a molecular scaffold to which other proteins involved in NHEJ can bind, orienting the double-strand break for ligation. The Ku70 and Ku80 proteins consist of three structural domains. The N-terminal domain is an alpha-beta domain. This domain only makes a small contribution to the dimer interface. The domain comprises a six-stranded beta sheet of the Rossman fold. The central domain of Ku70 and Ku80 is a DNA-binding beta-barrel domain. Ku makes only a few contacts with the sugar-phosphate backbone, and none with the DNA bases. But it fits sterically to major and minor groove contours, forming a ring that encircles duplex DNA, cradling two full turns of the DNA molecule. By forming a bridge between the broken DNA ends, Ku acts to structurally support and align the DNA ends, to protect them from degradation, and to prevent promiscuous binding to unbroken DNA. Ku effectively aligns the DNA, while still allowing access of polymerases, nucleases and ligases to the broken DNA ends to promote end joining. The C-terminal arm is an alpha helical region which embraces the central beta-barrel domain of the opposite subunit. In some cases a fourth domain is present at the C-terminus, which binds to DNA-dependent protein kinase catalytic subunit. Both subunits of Ku have been experimentally knocked out in mice. These mice exhibit chromosomal instability, indicating that NHEJ is important for genome maintenance. In many organisms, Ku has additional functions at telomeres in addition to its role in DNA repair. Abundance of Ku80 seems to be related to species longevity. Aging. Mutant mice defective in Ku70 or Ku80, or double mutant mice deficient in both Ku70 and Ku80 exhibit early aging. The mean lifespans of the three mutant mouse strains were similar to each other, at about 37 weeks, compared to 108 weeks for the wild-type control. 6 specific signs of aging were examined, and the three mutant mice were found to display the same aging signs as the control mice, but at a much earlier age. Cancer incidence was not increased in the mutant mice. These results suggest that Ku function is important for longevity assurance and that the NHEJ pathway of DNA repair, mediated by Ku, has a key role in repairing DNA double-strand breaks that would otherwise cause early aging. Also see DNA damage theory of aging. Name. The name Ku is derived from the surname of the Japanese patient in which it was discovered. References. This article incorporates text from the public domain PFAM and Interpro IPR 005-161. This article incorporates text from the public domain PFAM and Interpro IPR 06164. This article incorporates text from the public domain PFAM and Interpro IPR 005-160. This article incorporates text from the public domain PFAM and Interpro IPR 014893.